Ch2 series in Human Anatomy and Physiology. Chapter 2. Cell Structure and Function. Episode 11. The Process of Mitosis. If you enjoyed the video, don't forget to hit the like button and share it, to help us reach every medical and health professional student around the world. And of course, make sure to subscribe to the channel for more valuable content. Your comments are highly appreciated. Welcome to today's journey into the fascinating world of cell division. In this lesson, we will explore the process of mitosis. Before we begin discussing the stages of mitosis, I encourage each of you to revisit my previous episodes where we have covered how chromosomes are organized in a typical somatic cell and the differences between haploid and diploid cells. These can be found in Series 1 and 9 of Chapter 2 of this series. As a quick reminder, in a resting or non-dividing cell, chromosomes exist in the form of chromatin, a relaxed, thread-like material. Also, we have discussed that human somatic cells always contain 46 chromosomes, arranged as 23 pairs. Herein, one pair is inherited from the mother and the other from the father. During the S phase of the cell cycle, each chromosome of these pairs duplicates, forming two identical sister chromatids that remain attached at a region called the centromere. As a result of this duplication in the genetic material, the cell still has 46 chromosomes, but now contains 92 chromatids. To help us clearly visualize what happens during mitosis, we will use a simplified example of a mother cell with only 4 individual chromosomes. After duplication, this cell will have 4 chromosomes, each made up of 2 sister chromatids, giving us a total of 8 chromatids. This model makes it easier to follow the changes step by step. But keep in mind that the same events occur in real human cells with 46 chromosomes. The numbers are just larger, but the process is the same. Mitosis usually begins after the cell completes interphase, which includes the G1, S, and G2 phases of the cell cycle. During mitosis, a somatic cell divides to form 2 genetically identical daughter cells, each with the same number of chromosomes as the original mother cell. This process is essential for tissue growth, repair, and maintenance throughout life. For example, in a human somatic cell that contains 46 individual chromosomes, mitosis produces 2 daughter cells, each also with 46 chromosomes. In our simplified model, mitosis in a cell with 4 individual chromosomes will produce 2 daughter cells, each with 4 chromosomes. However, mitosis itself is typically divided into 5 main phases. These are Prophase, Prometaphase, Metaphase, Anaphase, and Telophase. This is followed by a separate process called cytokinesis, which physically separates the 2 daughter cells. This short video gives you a brief idea about the different stages of mitosis and the visual events that occur, which will cover them in today's lecture. Now let's go to talk in details about each phase of mitosis. As discussed before, our dividing mother cell model is a cell with 4 chromosomes. During the S phase of the cell cycle, these 4 chromosomes duplicate, forming 4 chromosomes, each with 2 copies or sister chromatids joined at a central point called the centromere. Now, the first stage of mitosis is the prophase. During prophase, the nucleolus which is responsible for making ribosomes disappears. Also, the nuclear envelope begins to break down, marking the start of visible cellular division. Meanwhile, 2 special organelles, called centrosomes, begin migrating to opposite sides of the cell. Form these centrosomes, spindle fibers start to appear. As we will see later, this spindle apparatus is essential for movement and alignment of chromosomes in the upcoming stages of mitosis. Prophase is followed by another phase called Prometaphase. Now the nuclear envelope is fully dissolved, and the chromosomes are exposed to the cytoplasm. Also, spindle fibers reach out and attach to the kinetochore, a protein complex located on each chromatid's centromere. You can clearly observe that kinetochore fibers connect each chromatid to a spindle pole. Finally, in the Prometaphase stage, the chromosomes begin to tug and jiggle, preparing for alignment. Therefore, this phase is all about connection and coordination, ensuring every chromosome is correctly paired with the machinery that will pull it apart. The next phase of mitosis is called the Metaphase, or the chromosomal lineup phase. At this moment, all chromosomes align neatly at the cell's equator, known as the metaphase plate. This is not a physical structure but an imaginary line bisecting the cell. In addition, each chromatid is now attached to both poles via spindle fibers. Importantly, a crucial checkpoint occurs in metaphase stage as we discussed in the cell cycle lecture. This is called spindle assembly checkpoint, SAC, and its role to ensure that every chromosome is properly attached and aligned. If not, division is halted. After metaphase, the cell undergo the great separation phase that is known as Anaphase. At the beginning of Anaphase, you can observe the 2 chromatids are held by a protein called cohesin. As magnified by this image, the first step in Anaphase is breaking down cohesin protein by an enzyme called separase. Now, each chromatid becomes an independent chromosome with its own centromere. Then, the mitotic spindle fibers undergo active shortening and pull the centromere region of each sister chromatid toward opposite spindle poles. In Anaphase, you can observe that chromosomes, at each pole, appear V-shaped under a microscope. This is because the centromere leads the way, with the arms trailing behind. Directly after Anaphase, you can recognize that half of the starting number of chromosomes appear at each side of the cell. This will represent the DNA content of each newly synthesized daughter cell. The last phase of mitosis is Telophase and is called rebuilding the nucleus. Now, at each pole, the chromosomes begin to relax back into chromatin. Also, new nuclear envelopes are formed around each set of chromosomes of the daughter cells. Moreover, the nucleolus reappears in each cell, signaling that protein synthesis can soon resume. Finally, the spindle fibers disassemble, as their job is now done. So, in telophase, the cell has essentially created 2 nuclei, each a genetic mirror of the original mother cell of 4 chromosomes. Although mitosis technically ends with telophase, the final act is cytokinesis represents the final split. Cytokinesis is also known as the division of the cytoplasm. In animal cells, a contractile ring made of actin and myosin forms around the center of the cell. This ring pinches inward, forming a cleavage furrow until the cell splits into 2. At the end of cytokinesis, each daughter cell receives its own nucleus, a full set of organelles, and the machinery needed to survive and function.